What a great honor to have Nicolas again with us. We talked last time, Nicolas, and we did something very interesting talking about the brain and if it was the origin or cause of consciousness. But since you did something extraordinary, first one in science, and you and your team showed that there is actually biophoton, ultra-weak emission of light from the brain in human. Do you want to tell us again where you work right now and a little bit your titles? And
Sure. Yeah. Thanks so much for inviting me back. Happy to chat with you. It's always a good time. Yeah. My name is Nicolas Rouleau. I'm an assistant professor in the Department of Health Sciences at Wilfrid Laurier University, an adjunct position at Tufts University in the Department of Biomedical Engineering, and I'm an affiliate scientist for the Allen Discovery Center at Tufts as well. I work on problems in neuroscience, a whole bunch of different things, but today we're going to talk about light because that's one of these things we've been working on in recent years.
yes, this last study, what was a breakthrough in science? Everybody talked about it.
Yeah. The major breakthrough here is that we've demonstrated in our recent paper in iScience that there is a relationship between brain activity as measured conventionally by tools like electroencephalography. There's a relationship between those kinds of oscillations and light emissions from the brain. We've known for some time that brains and other tissues emit light. We can go back to original work by
Gurwitch?
Gurwitch, right? In the if we go way back, we could look at Gurwich's original experiments. But if we go back 30 years or so, we've been looking at light emissions from the brain in slice cultures. If you take a brain from a rodent, cut it up into sections, and it's still living, and you support it with nutrients, and you place neurotransmitters onto those slices, the cells will react to the neurotransmitters, right? If you put a little bit of glutamate on there, glutamate is an excitatory neurotransmitter. You would expect the cells to depolarize. They do that, and you can measure that electrophysiologically. But the other thing they do is they emit photons. And the question is, what are these photons? Are they just byproducts of physiological activity, or do they have signaling capacities? And in recent years, there have been some researchers who've put forward some pretty compelling evidence that there may, in fact, be an optical modality in the brain. That is, in addition to action potentials, there may be light signals that are shared between brain regions that act as a carrier of biological information. And we ask the question, can we measure it? Is it related to known brain activity as measured by something like EEG, electroencephalography? And what are the implications of it?
Yes, and up to now, they call it ultra-weak photoemission. They were never totally sure that it can do something being so weak. It can be separated from the background information. And you really made it clear that it was something that has an function and it was separated from the background noise in the brain.
Yeah, if we focus in on the energies here, I think that it should be clear that even a single photon can activate cells in the visible range. It carries an energy equivalent of 10 to the minus 19th joules in energy. Even a single photon can activate one of these cells. And so, talking about ultra weak photons, which are many of them in the visible range, but also some in the UV and near infrared range, can they affect biological systems? The answer, based upon just an analysis of the energies involved, should be yes, of course. But the question is, how much tissue penetrance is going to be relevant to the actual signal transduction pathways? Like, does the photon have to go through anything, or is it simply moving to its neighbor cell, right? Like, if you have two cells right beside each other, photon emissions from one should readily affect the other. But how far is that photon penetrating? Can the light be guided? In recent years, we've seen some theoretical work by our colleagues out in Calgary, another Canadian breakthrough. We have evidence now of axons acting as potential light guides. So light can travel through tissues. Of course, it can do that. But the question is, can it bend and conform to the structures that are the brain rather than passing crudely through these structures, right? Can it be guided? And that would be pretty important in order for information to really have meaning in a biological context. And we know now that light can travel along these axons, and that should be something that we explore a little bit further.
So optic pathways, light pathways, I imagine microtubules, axon, what the other hypothesis to carry photons?
Yeah, microtubules are are certainly part of this. So are some other organelles, like mitochondria are part of the discussion as well. But it seems that at least the modeling suggests that myelin is probably pretty important. And myelin is, for those who don't know, the fatty sheath that surrounds neurons that we typically think of as an insulator that gives rise to the capacity for neurons to engage in what's called saltatory conduction, where the action potential moves from node to node, refreshing and maintaining that 120 millivolt depolarization as it moves along the axon barrel. But we think that actually the myelin could actually have optical properties that make it a great light guide. And that shouldn't be too surprising. I mean, we've seen evidence in recent years that the axon, as well as the myelin sheath, participate in a mechanical wave that accompanies the action potential. So there's a lot of biophysics happening here. In addition to the action potential, which is like in all the textbooks, everyone thinks of action potentials when they're thinking of neurons. There's actually a mechanical wave, and there probably is also a light, an optical wave that's occurring at the same time, all in parallel. Not only do we have electrical potentials, but in parallel to that, we have mechanical potentials, we have optical potentials. That increases the potential resolution of information by many orders of magnitude.
So when you say mechanical vibration, something like this, right? So saltatory means jumps. The myelin sheath are not connected as one. You have those space, the nodes of Randier. That means you have also saltatory movement of the photons between the myelin sheath. They would have to be saltatory also for the photons.
Yeah, you're right. There are gaps between them. So there's some very clever researchers who did something very clever where they impregnated these cells with silver nitrate. And they used this technique to basically photograph the insides of the cells.
I think Cajal, you were using that in Argentina.
Exactly. That's exactly right. Yes, it's typically used as the silver nitrate stain is the Golgi stain. It's right. Very famous. Yeah. But you can use this for a kind of autoradiography where you add the substrate into the cell. And if the cell is generating more light internally, so you have these cells in the dark, if it's generating more light internally, that should actually show up as pigmented grains of this substrate, like almost like the silver nitrate is capturing the light from the inside of the cell. And it turns out that if you stimulate those cells, which we know is coupled to more light emissions, you actually get more of these little pigmented grains inside of the cells. And these tend to cluster at nodes of Ranvier. So it's almost like wherever the myelin sheath meet at the nodes, there's actually a space there where the light is being emitted. Now, we don't know if it's because it's moving outwards or because maybe it's moving across the gap. Like there's a lot of unanswered questions here, but there seems to be a spatial component here that matters.
So we're talking now axonal transport because we're not talking about the myelin sheath. So it would be both in the axon and in the myelin sheath. And the fat tissue are known to carry and transmit photons? Medium?
Well, the modeling suggests that they do. We need more experimental evidence of this. And myelin is the body of a glial cell. It is membranous. It has multiple layers to it if you take the cross-section. So it's a complex structure. It's not just a blob of fat. The optical properties of that should be thoroughly investigated because some of the original modeling that we have now suggests that it probably could carry light in an efficient way that allows for an optical neural pathway.
So in the brain, just to review, we have a glial cells called the oligodendrocytes, and each cell have 10 to 50 extension, and every of the extension is a sheath that's going to go around an axon. So it's going to go around 20, 40, 50 axon like this. But if we think about heat, for example, then the heat will diffuse through the tissue without following the anatomical structure. We could imagine that maybe light doesn't have to follow this complex structure, just emit through, or maybe stay on the surface or not. About the axon, so we have, the perineurium, epineurium, endoneurium, and you have a lot of fluid there. And recently we showed that there's a CSF, cerebrospinal fluid, carried through those neural space, intraneural space. Could we imagine photon inside the fluid? Because we know photon can be carried by fluid. So can you imagine the photon carried by the fluid and released in the nodes of Ranvier between myelin sheath?
Definitely. I think that there's a lot of unanswered questions here because an optical pathway or many optical pathways in the brain would be very much unlike the synaptic conception of how brains communicate, would be pretty messy. So Suppose you have a light source. You can think of it as a candle. You could think of it as radiating outward. I could see some of those photons being carried by structures and guided, from A to B. But I could also see some of those photons passing through tissues. Some of them would be attenuated. So like there are certain wavelengths that are going to pass through less tissue than others. Some of these light guiding properties are going to be dependent upon wavelength as well. So there's going to be a lot of wavelength dependence. There's going to be a lot of spatial dependence, right? Whether a neuron is close to the surface of the head or deep within the brain, that's going to impact how far that photon travels and so on. I don't think we have the answers to all these questions, but I think there's many parallel sort of experimental questions that need to be asked to really figure out whether this is something that's biologically useful. We found that light that travels outside of the head actually has some relationship with neural oscillations at the level of the cortex. So let's say that at a bare minimum, the top, let's say 5 millimeters, this cortical region is related to photons in some sense. We have the evidence to show that the information structure of these two signals are related to each other. But what about deeper signals? We don't know. What's happening at the microscopic scale? What's happening at the level of axons? And how are glial cells related to this? This is a field that's now sort of blown wide open where we need people to address these questions, and we can't do it on our own. There needs to be a community that comes together with an interest in ultraweak photon emissions in neuroscience.
And infrared has been shown to go a few centimeters through the skull. And again, it depends where there's some different of thickness in different part of the skull. But so we're thinking at least a few centimeters, right, to being conservative. Yep. Let's stay with transmission of light. So we're talking about axon, myelin sheath, microtubule. Did we show there is some transmission in the center of those microtubule, the cytoskeleton?
Yeah, there seems to be some light guiding properties to the microtubules as well. There are others who are working on these questions, and they probably work with mitochondria. So like the two of them together, which are abundant in axons, but are also found throughout the cell and obviously in other tissues.
All the microtubules in a cell normally are centered around the microtubule organization center. It's my favorite structure, diplosome, centrosome, which are located close to the nucleus. So there is some talk about the DNA creating biophoton. So that would be a third source from the DNA inside the nuclear membrane. We could have photons here going to the microtubule, going to the mitochondria, or vice versa, from the mitochondria to the nucleus.
Yeah, there's a lot of hypotheses about where the photons are coming from. Most of the attention has been on metabolic activity from mitochondria, but there are others who've suggested processes at the level of the membrane. Others associated with DNA, looking at things like DNA replication and how that impacts photon emissions. But the reality is, like, a photon can be emitted from any system that undergoes an energetic change. And anytime you have that energy change, you can have the emission of energy in the form of light. Sometimes that's in the form of heat, but other times it's visible wavelength. If we're looking for a single source, we're going to end up disappointed. There's probably multiple sources, but then again, we can probably rank order them in terms of which ones are contributing to the biggest signal. I should say that it may be that certain subsets of the spectrum that are being emitted from a cell are going to be preferentially related to certain kinds of organelles or certain kinds of molecules. If we look at different kinds of molecules in the body that we know are related to autofluorescence, like flavins, serotonin precursors like tryptophan, these are molecules that we know have interesting optical properties to them. And if you hit them with light, they will re-emit photons. There's a lot of photoreceptive molecules in the body, and they're ubiquitous, like these proteins, these neurotransmitters, amino acids, things like porphyrins, all sorts of molecules that are going to have these optical properties. So we have to look at all of these and really tease out where the contributions are coming from.
Yeah, I have a list of about 20 of those somewhere. Origin of the biophoton in the cells, from lipid peroxidation inside the respiratory chain of the mitochondria, the oxygen decay, the chromophore proteins, coenzyme Q10, melanin. All the tryptophan, tyrosine, phenylalanine was the main source of biophoton right now. We still think most of it is from the reactive oxygen species created by the mitochondria.
Yeah, we think it's mostly related to metabolism. Right. As soon as you relate something to energy, there's all sorts of confounding factors that come into play. Like, of course, it's going to be related to energy because anytime the cell does anything, it's going to be using energy to actively do that. And so, of course, energy production is going to go up when a cell is firing an action potential. Of course, energy production is going to go up when the cell is engaging in protein synthesis and all these sorts of things. Like, of course, energy is going to be related to the ebb and flow of the physiology of the system. The system is drawing power from its energy source. Too much focus on the idea that, well, yes, it's metabolic processes that are giving rise to these photons. I think it leaves you with too many questions, right? It leaves you with, well, is this a byproduct or is this a genuine signal?
So when the system raises energy, is when the energy go down a little bit that at this moment you release a biophoton.
Yeah, there's also interesting spectral properties that are relevant. So the frequency of emission of those photons changes. It's not that they're all being emitted in a very predictable sequence. There seem to be bursts of these photons. There seems to be nonlinearities in terms of emissions that are probably relevant and actually allow you to classify cancer and non-cancer cells, for example. So currently, yes, I think there's a big focus on intensity, which is really no different than any other imaging modality. When we look at, for example, like CT scans or, ultrasound, intensity is obviously the first thing that jumps out, but pattern is something that we're also very much interested in and seems to matter for these systems. Like, what is the emission pattern and what are the wavelengths of the cells? And for that, you need to actually apply filters to your measurement devices. You can't just take all the photons. You have to only look at a subset of the photons, which may be emitted in a totally different pattern than another subset.
So a few people showed a rhythm, a beat, a pulse in those biophotons. And so you would try to measure those pulses to see if there's information in that rhythm?
Yeah, we have done that, and we've largely focused on low frequency emissions because of the limitations of our instruments. But if we increase the resolution of the instruments, we could look at much higher frequency emissions in terms of the pattern of emission. And one of the big questions right now is, what is the relevant timescale? Because if we're talking about something on the scale of action potentials, we're talking about milliseconds. But if we're talking about something that's much faster than that, that potentially is quantum or is something that's happening many times per unit action potential. Now we're talking about something that's potentially at the nanosecond scale, right?
That's fantastic. We work with a rhythm, 0.1 hertz. This is a very important rhythm for health. It's the rhythm of the lymphatic contractions and baroreceptor. Some hormonal secretion are around 0.1 hertz. If I remember, you had some important emission around this 0.1 hertz.
Yeah, we did notice emissions under 1 hertz. I think they were at 0.1 hertz. And that was one of the discriminating frequencies that we noticed in terms of light emissions that could distinguish brain rhythm from non-brain light signals. So if we had a photomultiplier tube in the room measuring background light, and these are pretty dark rooms, but there's going to be light, ambient light. It was that frequency that was the major contributor to being able to discriminate between a brain signal and a non-brain signal, which was really fascinating.
Yeah, that's an important rhythm. There's some studies about 0.1, 0.15. It's a very important rhythm, and that's something you can attune your breathing, can attune many things, and that's going to really calm you down. And when you're calm like that, you're going to go more parasympathetic and regenerate your tissue. This is something that we can use with our hands, with our breathing, to bring health in the system.
That's so interesting. I wonder if we could. You're giving me ideas now. So if we could, that's why these conversations are great. So if we could take that 0.1 hertz photon rhythm, let's call it, and we could engage that with a brain-computer interface for biofeedback. That would be super cool. I mean, potentially we could show that as you either increase that signal or decrease that signal, other health markers are going up and down with that sort of deliberate, intentive process.
I sent you some article about this, but this is a rhythm you find in the intestine for contractility. You find that the lymphatic rhythm, some brain rhythm, baroreceptors, again, hormones, secretion, and I expect some kind of coherence at 0.1, 0.15. And it's a very simple application. Just beating that, making a beat like that with a machine with a nice frequency, a red frequency or something like that will really add to bring back people into more parasympathetic and a place when they can rebalance their autonomic nervous system, their their hormonal system, blood pressure?
Yeah, and I think that's the other piece of this, which is, can we use our knowledge of ultraweak photon emissions and brain optical networks to influence brain health? There's a whole field that existed before any of this, which is photobiomodulation, which is already showing that, if you pulse light on other tissues, but the brain as well, you change physiology. And that should be unsurprising to most people because cells have evolved in an environment with a lot of light signals. We have a nuclear reactor in the sky, which is in many senses the origins of life. And so it should be unsurprising that cells are responsive to light. In your brain right now, you have cortical neurons, which are not retinal cells. These are not visual cells, and yet they express opsins, which detect light. And so potentially the 0.1 hertz rhythm could be important, married to certain kinds of wavelengths. Perhaps some wavelengths work and others don't, but these are all empirical questions. We have to see if, when you expose people's brains to these light patterns, if in fact it changes all these health markers.
Yeah, let's talk about wavelength and frequency, because the sun only emits 600 nanometer to 3,100 nanometer, and there's a lot of important frequency lower than this. And you didn't really look for different type of light frequency in the study. I imagine in purpose to simplify the results, or?
Yeah, so the detectors that we used for the study have an intrinsic wavelength range. So I think it goes down to around 400 nanometers to around 700 nanometers, let's say. So it's in the visible range, but there's obviously going to be some contribution from near UV and near infrared. We kept it open deliberately because we don't really know what we're looking for. We know that photon emissions are present, but we don't know which subset are going to be relevant to brain activity and cognitive tasks. And so we looked at the whole spectrum. But of course, you could do a similar study, and this is one of the things we're doing now with filters and with other instruments that have different spectral ranges to look at these different subsets. And we think that those are going to be highly relevant.
Perfect. Yeah. One more thing, talking about transmission of light outside of the cell, what do you think about the PVS, the primary vascular system?
Well, like you said earlier, I mean, once we're talking about liquids and we're talking about these sort of fluid dynamics, and they're going to have different refractive indices and things like this, I think that they're all relevant. We just don't really know how it's going to play into biological signaling. Like, of course, it's going to have a physical impact. There's no doubt about that. But we don't know the answer to a lot of these questions, and so they have to be empirically tested.
Another crazy question. What about my photoemission influencing your photoemission? Me as a manual therapist, how much do I influence with my photoemission? And their photoemission giving me information. There's a whole other field there. And we've done that. People are working on the Qi from the Chinese Qigong masters. And
I think one thing that's super interesting about that idea would be sort of reconceptualizing disease transmission. If indeed these ultraweak photon emissions are related to physiology, that means they should also be related to pathophysiology. And if they're related to pathophysiology, the question is, if they carry biological information, and we know these photons are getting out of the head, if they can get out of the head, they can go into a head. So if they're coming out of a head, is it possible that, especially in darkened environments, especially in environments where we don't have, overhead fluorescent lamps, maybe around the campfire, is it possible that information, biological information from my brain is transmitted to someone else's, or indeed information from someone's heart or from another organ, which, by the way, all emit these ultraweak photon emissions? Could those affect my physiology? And I think there's already evidence to suggest that they do. I mean, photobiomodulation is the extreme example where you're using much higher intensity light, but there are examples of studies where people have, the way that this is explained is, we're doing Reiki practices or other kinds of traditional and alternative medicines. But suppose that simply putting your hands over someone or simply being close to someone allows for you to transmit some biological information to them through these optical pathways that we're just not currently, very familiar with. I think that there's a whole field here potentially that could benefit therapeutics, but we also have to think about how this implicates the idea of like different methods of transmission of disease. This would be disease caused by neighboring cells that are dysfunctional, that are not in your body, that are in the bodies of other people, but are influencing your body simply because that signal is not a molecule. It doesn't need to be bloodborne, and it's not really airborne. It travels between us as light. This is, of course, speculation at this point, but it seems plausible and can be empirically tested. It would have us rethink medicine, but it would have us rethink things like architecture, building hospitals, for example. Do we really want to have people in rooms beside each other with a sort of curtain between them, or do we need to optically isolate people in the same way that we currently chemically isolate people, right? We don't let people just bleed on each other or breathe on each other.
So that makes me think about the applications. Are we going to have something more practical, that photo multiplier that we have put people, in a very dark room? I mean, practically, how are we going to, do that in a hospital, even in an office?
Yeah, I think there's a number of ways to do this. It becomes a little bit complicated with the brain because, well, for the same reason that it's been difficult to access the brain with any technology. It's inside the skull. It's intrinsically invasive to get close to it. But then for other tissues, like if we're just looking at, the skin, which is the most superficial organ, little enclosures, potentially a kind of box that your arm goes into, or, maybe a cuff or something that can be light tight, at least locally. These would be useful wearable technologies that you could use to monitor UPEs at the surface level. Deeper tissues are always going to be a bit more difficult, but these surface tissues, I think that there's definitely an opportunity to have something that's really practical that you could have in an office or have in a clinic without turning all the lights out and putting padding on the windows and all these things.
Yeah, we don't know still what we're looking for pathology. We're looking for photon emitted by the vasculature, by the skin itself, by the neutrophil inside the vasculature, by deeper nuclei in the brain. I mean, we don't even know exactly what we're looking for.
Yes, there's a lot of noise in the system. We don't know what is the relevant noise and what's the irrelevant noise, or rather the signal. And we need to parse all these signals to figure out what is the determinant signal feature that allows us to predict what will happen next in the cell culture or in the animal.
So what are you exploring right now?
Yeah, we're looking at expanding on this technique we've developed we've called it photoencephalography. So it's like electroencephalography, but for photons. And we're expanding on this technique by looking at filters, number one. And number two, looking at higher spatial resolution to see if we can source localize photons in three-dimensional space. But we're also looking at things like how different wavelengths and different patterns of light can be transmitted by biological tissues. And so we're doing a lot of different things, trying to answer different questions, but a lot of what we're doing is aiming toward refining this technique, making it more useful. We know we can discriminate brain from non-brain. We know that it's related to some of the things that brains are doing that we already knew they were doing. And we know it's related to function in terms of cognitive states, but we don't have enough information to really make it something that's practical for, clinicians, at least not yet, but we're getting there. And by answering some of these small questions, we'll get to the big question, which is, what does light emission from the brain really mean? Is it a byproduct? I think it's very unlikely at this point. Or is it a signal that we don't yet understand that could be quite useful once we do, and once we decipher the messages that are all around us?
Photoencephalography, how deep do you go?
We don't know. I mean, we know that light can pass directly through the brain and skull, and we've only measured on the outside of the skull, and we've related those UPEs to electrical activity that we know is coming from the cortex. That's the EEG, typical EEG activity. So we know it's going to be related to those first few millimeters of the brain. But we also know that the deeper structures of the brain are emitting UPEs. We know this from the slice culture work with rodent brains. Now, there's a couple questions here. What are the deep UPEs doing? Are they influencing the more superficial UPE emissions? Is there a kind of chain reaction effect there that's happening? Are they interdependent? Are they independent? We don't know. But currently, we understand that at least the upper layers of electrical activity of the brain in the cortex are related to these UPEs. We suspect that with refinement of the technology, we'll be able to relate it to these deeper brain rhythms as well.
But at this point, you don't discriminate if the EP are coming from the hippocampus or the thalamus. You're not able to do that yet. You just get...
Not yet, no. We need higher spatial resolution. That's one of the things we're working on, but we also need to validate that against existing technology.
So right now, basically, the pathology you're working on cancer, are you working on aging maybe?
We're not working on aging per se. We're sort of interested in these sort of fundamental questions. We're not taking a kind of health approach to it, although the cancer work does. But some of the brain work we're engaged in is much more focused on, what are the fundamental mechanisms here that are allowing this to happen? And then those can be applied to individual pathologies to really kind of help in those clinical contexts.
Now, when we talk about heat, for example, heat we can say is infrared, and the heat produced by our metabolism diffuse everywhere. I don't know if you had specific pathways for heat. So some part of light could find easier way than electric signal to go through the tissue. At least some frequency, like infrared, will propagate everywhere. So not every type of light needs specific optical pathways.
Right. Yeah, you could think of it as analogous to the endocrine system. It's not that there's a specific pathway that allows you to transport the hormone directly to the tissue. It's that there are certain tissues that are receptive to those hormones. And if the hormone goes everywhere, if it diffuses throughout the body, in this case through the circulatory system, then the areas that are receptive to it will become activated, and the areas that are not receptive to it won't become activated. It could be the same thing for light and for heat, where the signal diffuses, yes, and maybe it does so evenly, maybe it does so unevenly, but it diffuses. And only the areas that are receptive to that light, either the wavelength or the pattern, become activated.
Now, almost all objects in a room emit infrared, right? There's a bit of heat. What kind of spectrum of light an object emits? Do you check into what frequency an object can emit?
Yeah, it's going to be dependent on each object. There's always going to be a spectrum. It's never going to be a very punctate peak. There's always going to be a spectrum. We have more photon emissions than inanimate objects, unless, of course, it's like a steel ball that you've put on on a flame or something like that. And of course, if you take an animal that was alive and then recently died, you'll notice that the photon emissions also decrease. You can get an initial increase in photons upon death in cells or in organisms, but ultimately the amount of photons does decrease over time as the system dies and cools, I'll say as well.
Also, did you see this article? I think it was from Wuhan, if I remember. They talk about the red shift.
Yes.
Red. So through evolution of animals, humans have more and more red shift.
That's Jai-Pei Dai's group, and he's one of these pioneers in this area as well. And yes, he found that if you take animals with different cognitive capacities and you arrange them by cognitive potential, so if you take like pigs and rats and cats and dogs and humans and elephants, and you were to seriate them and actually have really high cognitive potential and then less and less so. And of course, those are open to all sorts of operational definitions. Like these are things you have to think about critically. But if you do that, and then you look at red shifted light versus blue shifted light, you'll find correlations between these things. And so the question is is the wavelength reflecting the cognitive capacity, or is it reflecting the energy, which is also reflective of the cognitive capacity for reasons of biocomputational efficiency? A system that is less efficient, computation by computation, will produce more heat, will produce more energy, will require more energy to get the same output. There's questions here about efficiency and things like this, but there's also questions about are there parts of the system that really just are there in the high cognitive potential animals that are not there in the others, that are giving rise to these different emission wavelengths? And we also know that, as an animal ages, we do see shifts in the wavelength of their light emissions as well. So the senescent animals will show more, I believe, of the bluer wavelengths and ultraviolet wavelengths.
So a baby is more red, and with age we get more and more blue and purple. So that means if we really evolve, we're going to continue to get more and more red. We can expect us to be glowing orange very soon.
Right. Yeah. Actually, and that's a question we do get asked often, is how does this relate to auras? The experience that people have, that some people have, of seeing a glow around others, particularly in darkened settings. Right. And I think there's something to this. I mean, like I said at the very beginning, it doesn't take many photons to activate a rod or a cone, right? It takes one, exactly.
One photon is enough to activate the retina in a living human, yes.
Exactly. So our study demonstrated that there are photons coming out of the head. We know this is also the case for other tissues of the body. And so the light is there. And we know that the retina can detect this. We know that the cells are able to detect it. The only question is the intensity sufficient to be registered as a visual experience? And we already have people reporting the visual experience. So now we just have to take this seriously, which a lot of scientists don't do. Take the experiences of people seriously and test the thing. Put people in a room, have them in darkened settings, different background ambient light settings, and see if you can measure the light emissions and if those light emission patterns correlate with the experiences that people have about other people's potential aura, a perceived light coming off of others. We have experiences going way back of people seeing flashes of light upon death when their family members die. We have experiences like auras. We have other kinds of visual experiences that are associated with near-death events. These things are often written off as people under stress, anomalous experiences, or they just don't exist. Take the experiences seriously and test it. We now have the technology to do it, so let's test it.
I actually see auras when I work. I use it. It's not something I can control all the time. And I think when I talk with other people, other therapists seek slightly different things. Depends of, our receptors, sensitive to receptors. And I guess meaning we put, so there's an intent, how the intention is going to change my perception. If I am used to put, like I see red, there's an emotion of anger there on top of the tissue. Depending of my way of reading that, I already have a preconception of something. I guess that's why people have different reading. There's also different dimensions, different depths. People are looking at chakra, people are looking at the emission from the skin. So we would have to kind of put some kind of standardization. We all agree to look at this or that, or.
But the experiment allows us to answer this question. So there are some people who would be tempted to dismiss this and say, well, look, if you have two different people looking at the same person and seeing two different colors, like one reports green, the other reports orange, for example, you might say, well, look, you're demonstrating that it's not subject to inter-observer agreement. So there's two people who are both looking at the same thing, they're reporting different things. You haven't done the experiment. So what you need to do is you need to actually have the same person generating the light, both people reporting their experiences. And then at a later time, or when something has changed, right, you can experimentally manipulate the situation. The next time they report the light, do their responses track each other? So suppose that every time the person who's seeing green starts to see the color as being a little bit more blue, the person who's seeing orange starts to see the color a little bit more yellow or green.
Yeah.
They're both being blue shifted, right? So is there a relationship between the shift in the wavelength? That's number one. The second is maybe there's a sort of category thing here where they're both being perceived differently, sure, but they're being perceived differently reliably. So every time I see red, you see blue, every time. And every time I see green, you see orange. That is a systematic relationship that is much more informative than simply saying, well, we're seeing the exact same thing. If there's a systematic relationship between the reports by two different people, you are in fact detecting an objective referent in the environment. That is classic psychophysics. You are seeing something, but you're perceiving it differently. And that would be relatively unremarkable in the history of psychology. People perceive things differently all the time. But are the misperceptions systematically related? That's really the critical issue.
I love that. Last time we talked about the question if the brain, the source of creating consciousness. Well, since then, I don't know if you saw that man that was like an average guy. He went to do his MRI and they found he has basically no brain left. He has a whole cavity of liquid and a tiny sliver of cortex around it. And he never had any medical problem. Nobody could answer that question. Why could he function pretty much normally? I wonder is biophoton, coherence, quantum principles could multiply his capacity like that, or if the biophoton can give an answer to this.
That's great. I think that's super interesting. These people are very sensitive to brain injuries. Like if they get what would be equivalent to a mild brain injury in an individual with normal brain structure, this would be much more severe for someone who has this sort of squished phenotype. That does suggest that structure is important. So like there's something about, even though it's squished out to the periphery, there's something about the structural integrity of the thing that does matter. But you're right, like maybe the conduction of electrical potentials is less relevant than photons moving out throughout the brain.
What I saw was such a small parenchyma, a small layer of cortex. I was really shocked. So it could be just the cells compacted, or you must have some type of necrosis, some less brain cells there.
No, you're right. There has to be some. So if you take another disorder characterized by widening of the ventricles, if you take like schizophrenia or dementia, right? Like if you take Alzheimer's disease, the reason why you have widening of the ventricles is because the tissue's dying off and it's becoming less voluminous, and the volume gets occupied by ventricular fluid, and things expand because there's just less pushing on it and so on. In this case, you have pressure. It's not an absence of tissue. It's actually just too much pressure and the thing moves out. I haven't looked at the histology, and I wonder if the histology preserves, like if it's really just compacted, but you're right. There has to be less volume there.
And also, the sulci, the valley inside the brain, we know something called lysencephaly. When you get dementia, everything is flattened. And there is probably light connection. This is my subjective perception. When I work on the brain, those deep sulci, deeper, I think that brain is 2/3 hidden.
Yeah.
The cortex in the cerebellum is 9/10 is hidden in the deep fold of the cerebellum. And there is a lot of exchange for me, what I feel in that space has to be more light exchange. And those connection doesn't happen. He's supposed to change something, but this man was on a microscopic level. He couldn't see any dementia, any retardation, any IQ problem.
Yeah, I think like a big takeaway from this kind of case study is something that I'm interested in my own work, which is the brain is an incredibly plastic organ. And if you preserve the inputs and outputs, if in fact there are sensory data coming to the cells and the cells are connected to one another and they can output to pathways that allow you to move your body, right? Like the motor strip, for example. So if you can maintain the sensory motor loop in the organ, and you do so from a relatively young age, the system will preserve its function. It is sufficiently plastic to rearrange its network structures to still solve the problems that it needs to solve.
Perfect. Nicolas, I know you are really busy. I don't want to take all your time. That was a fascinating discussion, as expected, and hopefully we'll continue after your next amazing breakthrough publication on biophotons. Thank you.
Can't wait to do round 3 with you.